by reversible covalent modification. For many enzymes, activity also is regulated by reversible covalent modification of one or more amino acid residues in enzyme. Over 500 different types of covalent modifications have been found in proteins. Common modifying groups are shown in the figure 6.35 and include phosphorylation, adenylation, methylation. The small protein is attached to lysine residue in the target protein, flagging it for the proteolytic destruction. The modifying group are usually attached to a regulate enzyme by a separate modifying enzyme. A metallase in case of methylation, introduction of modifying group alters the local properties of enzyme, which typically undergoes a change in conformation and activity. Introduction of a hydrophobic merstral group triggers the association of the protein with the membrane. Is the most important type of covalent modification and is discussed further in subsequent slides. Regulation by reversible phosphorylation, muscle glycogen phosphorylize. It is estimated that one-third of all proteins in a eukaryotic cell are phosphorylated, and one or often many events are part of virtually every regulatory process. Some proteins have only one site, whereas others have several, and a few has dozens of sites for phosphorylation. The attachment of the phosphoryl group to specific amino acid residues of a protein is catalyzed by an enzyme called protein kinases. In this reaction, typically the gamma phosphoryl group from a residue triphosphate, usually ATP, is transferred to a particular SCR, THR, TYR or occasionally HIS residue in target protein. Removal of same phosphoryl group from the protein is performed by enzymes called protein phosphates. The introduced phosphoryl group can cause major changes in the conformation of the modified enzyme by virtue of steric effects, hydrogen bonding and ionic interaction to neighboring residues. This can lead to effects on substrate binding and catalysis.